Hi, everyone. We're going to get started with the spotlight session. So I think 8-ish talks that are 3-minute long. If you're presenting a spotlight in this first session, please have a seat on the front row so you can all come when your turn is up. So maybe I can get the first presenter.
So good afternoon. My name is Francesco, and I'm here together with my colleague Vamsi to present our work on full-length ribosome density prediction from sequence and graph-based representations. So the ribosome density prediction tasks involve using some AI model on sequences, and we saw today already many of those, to predict, starting from an mRNA sequence splitted in codons, the codon-wise ribosome density. So the ribosome density is telling us where the ribosome is stalling in the sequence. And knowing this is very useful because it leads us to better understand translation elongation. And in turn, this can help us in doing, for example, codon optimization, in which we make the ribosome stall at the position that we want. So our work distinguishes from the previous one, since we are trying to consider both the mRNA sequence and the mRNA secondary structure. So if we consider our codon-wise sequence, essentially we are adding structural neighbors, or jumps, between the sequence corresponding to the edges in the secondary structure. So our method is named RiboGL, and is a combination of a graph and recurrent neural network. So we learn both graph embeddings and embeddings through the recurrent neural network. And our method performs best on our dataset of mouse liver ribosome density, ribosome profiling data, against the current state of the art that is named RiboMimo, that is just a recurrent neural network, so based only on the sequence. And our ablation study showed that still the sequence is giving most of the information. And this, I think, is a very interesting task since we have essentially a graph overlapping a sequence. So these kind of tasks are very interesting, I think, widely represented in biology. So I think our work can lead to similar results in other tasks. And so while our method performs similar to a sequence-based model, we can see from the interpretation extracted from CAP2 that we can get longer interactions, so longer interactions. And so if you consider, for example, the sequence above, where the A site is AAG, so the ribosome stalling we are considering, and we can get where is the most important codon leading to this prediction, we can check the distance. And when we do this on a BiLSTM against our model, we can see that the BiLSTM is not able to capture longer age dependencies as our RiboGL is able to do. So we provide also a visualization tool to see the result at the mRNA sequence level, and that is available on our GitHub repo. And we are waiting for you at the poster 1 to discuss some future direction on graph-based modeling for ribosequencing data. Thank you.
So hello, everyone. Our group worked on investigating data leakage when using protein language models. So I'm sure you've already heard of data leakage, but I will give you a quick recap. So data leakage happens, for example, when you use a pre-trained model, for example, a protein language model for a downstream task. And then the pre-trained dataset and your dataset might share samples. And if these samples, which the protein language model was trained on, are also in your test set, which you might not know about, this could result in unrealistic performance in the evaluation later. We decided to test this on thermostability prediction from ESM2 protein embeddings on the Meltome Atlas dataset, which contains proteins and their respective melting points. And we tested this on 2 different splits. One was the flip split, which doesn't consider the protein language model pre-training. You can see right here that the protein cluster C is contained in the ESM2 pre-training set. So that could lead to to improved results, which are due to data leakage, not due to your model being generalizing better. Then we also created our own split, which is called the EPA split, which removed all proteins of clusters that ESM2 was trained on from the test set. So that split is data leakage free. And we tried this out for a number of variants of ESM2 and also ESM1B. And we see a significant increase of 11% on average. And the experiments on the flip split outperformed the experiments on the EPA split on all architectures. And what we conclude from this is that data leakage seriously distorts the measured performance when you don't think of it when constructing new dataset splits. And we also think that other downstream tasks are affected, but this probably needs to be proven with further research. So I hope this reminds everyone to check their dataset splits when using pre-trained protein language models, because this problem doesn't really get noticed if you don't know about it. And if you want to discuss this further and are interested in additional information, you can look at our preprint or at our poster number 2. Thank you.
Hello, everyone. Let's meet PathoLM, the model we built for identifying pathogenicity from DNA sequence using the genome foundation models. So first, what is pathogen? So pathogens are organisms like virus, bacteria, fungi, those spread disease among human and animals. So by identifying pathogens, so there are species for whose some strains are pathogenic and some strains are not pathogenic. So our objective was to build a model that can well distinguish the pathogens from the non-pathogenic sequences. So our key focus are limited to the most clinically important escape pathogens, including École, Enterococcus, and some other species. And for virus, we took coronavirus, norovirus, influenza virus, and those from NCBI and Petri datasets. Okay, so what are the prior works on pathogen identification? Can be categorized in 2 parts, alignment-based methods and ML-based methods. So the alignment-based methods are computationally very heavy because they relies on the known database and sequence search. The ML-based methods resolves this problem by a large margin, but they need manual data curation and feature extraction. So what we did, we used pre-trained genomic large language models to fine-tune for pathogen identification task. And one of the important part of our project was to manually curate the pathogen data sets because it's limited and not very well distinguished because which strains are pathogenic and non-pathogenic. This decision was very important. And we fine-tuned nucleotide transformer version 2, 50 million parameter version, and solved the problems of minimal level data sets using the pre-trained knowledge. So why nucleotide transformer over other DNA language models? So for answering that questions, we experimented with zero-shot and few-shot learning. So as you can see that nucleotide version 2, transformer version 2 performs moderate, but DNA-BERT-S performs very good on zero-shot learning, but the learning curve for nucleotide transformer version 2 was higher than the other DNA language models. So that was one reason, and another thing was it is computationally less extensive than the DNA-BERT-S. So one of the goal was to build a lightweight model for pathogen identification. And we benchmarked with other models, DCI Patho, and we outperformed using in a large margin for all the evaluation metrics. And we also extended our PathoLM for species level classification, and as there is no model built dedicatedly for species classification, we compared with traditional machine learning models and deep learning models and showed that our model outperformed all of them. And also, we also showed the performance variation for varied sequence length, and the blue line you can see PathoLM SP outperformed for short read and both long read sequences and outperformed all other methods. Yeah, for more details, please visit our poster presentation. Thank you.
Hi, everyone. I'm Adam. I'm a PhD student with Charles Danko over at Cornell, and I'll be giving you a quick introduction to some of our work on training deep learning models on personalized genomic sequences. So I'm sure all of you are aware, there have been some pretty spectacular advances in sequence-to-function models over the last several years. But one of the main challenges, I think, to these models is that predicting the effects of non-coding variants remains quite challenging. And this is highlighted by these two recent papers benchmarking the Informer model. And one potential solution to this problem is to incorporate genetic variation into the training data sets. This strategy has been used by a number of different groups, many of which are represented in the audience today. And so our sort of approach to this was to train the ClipNet model, which I think you all heard about this morning. So what this does is it's a CNN-based model that predicts the transcription initiation signal around enhancers and promoters at single nucleotide resolution. And the way that we trained this model is we used matched genomic and ProCap data sets from about 60 genetically distinct LCLs. And in our paper, we showed that this model does reasonably well at predicting both across loci and across individuals. And so this result sort of naturally raises the question of how many N do we actually need? What's the relationship between the number of individuals in the training data and the performance, particularly on variant effect prediction? And so what we did was we subsampled the number of individuals that we use in the training data set and then evaluated both the cross loci and the cross individual performance. And to cut a long story short, what we found is that the more individuals you feed into the model, the better it performs at both tasks, which seems pretty sensible. Another issue that we wanted to tackle was whether the sort of benefits that we gained from training in this sort of variant-aware fashion can be retained after fine-tuning to a novel cellular context. So what we did is we took our ClipNet models that were trained on all 60 individuals and then fine-tuned it to a ProCap data set that was collected in K562. And then focusing specifically on the variant effect prediction tasks, we then benchmarked our model as well as ProCapNet, which is a similar sort of CNN-based model, as well as Informer, on predicting the effects of variants in a K562 MPRA. And what we found is that our model outperformed both of these two models, which I'll note were natively trained in K562. And so I'll finish by arguing that training models on genetically diverse data sets can improve variant effect prediction. Thanks, and I think based off the order, I'm poster number 4, but not entirely sure of that yet.
Sorry, we don't really have a strict poster ordering order. We'll see, but hopefully people can find each other. It's not that big of a poster session.
Thank you. Hi, everyone. My name is Ray. I'm a PhD student from Kristen and Leslie's lab from MSK. Today, we're going to introduce Hi-C2Self, a self-supervised Hi-C contact map denoising model. So this is the general framework. It's very simple. There are 3 different components. First, since this is a self-supervised model, we have the input matrix as this low-coverage input matrix. They're used as both the input and the training target. And then the second, we use a mask to achieve the self-supervision training. And then at the end, we design this 2-channel output, and then so that we can use the negative binomial loss so that the model can directly train and run on the row count matrices. We explored the model in 4 different use case scenarios, 2 on the bulk and then 2 on the single cell. First, on the bulk library denoising, we can see on the top is the low coverage input data of the Hi-C, and then this is the Hi-C2Self recovered results, and then this is the deeply sequenced model that the deeply sequenced library that the model has never seen. And then the second, we also explored our model onto micro-C data at 1 KB resolution. And then we can see on the top, this is a very lowly sequenced micro-C library. And then in the middle, we can reconstruct some of the structures. And then at the bottom is a deeply sequenced library that the model has never seen. We also cause significant interactions, and then showing the ROC curve over here. In addition, we also tried the model on trying to reconstruct some of the structures for the single cell Hi-C. First, for many of the single cell Hi-C, they actually have the co-assay which has for example, they have a taxi, a single cell ataxic and a single cell Hi-C. We can first bind kind of cluster the cells based on the ataxic, and then try to explore and reconstruct the structures on the pseudobulk of the Hi-C. So here, for example, we show a 50-cell pseudobulk at 10 kb resolution around the local gene, and then we can successfully reconstruct this unique structure in the cell stage. And then at the end, we also try on the single cell data. And then here we are showing at 50 kb resolution. Here are 2 random cells from one cell from a lowly methylated region from a gene, and then this is a highly methylated cell. And then we can see the model can reconstruct the different structures. We also try to cluster cells based on their local structure around this gene, and then color by the methylation level. The 3D structure reconstruction shows consistent pattern with the methylation. And then here is the model. We have 4 different applications. And then for more, please go to our poster number 5. Thank you.
Hi, everyone. My name is Aniketh, and along with Daniel and our advisor, Nyla, we have been working on conditional diffusion models for generating stable mRNA sequences. So as we all know, mRNA therapeutics have been a very promising field of research, but currently they're held back by certain biophysical properties such as stability. And current approaches to optimize for stability mostly concentrate on optimizing individual components of the mRNA sequence, a common example of this kind of approach being codon optimization. But they seldom consider the full sequence during this optimization. So to alleviate this sort of problem, we use diffusion models, which can actually look at the full sequence during sampling. So what we do is we train conditional diffusion models, which take in the full mRNA sequence as an input, but we condition on 2 types of stability quantifications, half-life and minimum free energy. Half-life quantifies the rate of decay of an mRNA sequence, and minimum free energy, or MFE over here, is like a thermodynamic measurement of the mRNA sequence's stability. By conditioning on these properties during training, during sampling, we're able to provide a desired value for each of these properties and generate corresponding mRNA sequences.
So generally in mRNA sequence design, we're interested in actually fixing the amino acid sequence for a desired function. So we reflect this in our analyses. This is showing 65 randomly sampled genes from our training data set, and then we fix the coding sequence and then use our diffusion model to optimize the 5' and 3' untranslated regions to make more stable mRNAs. On the left, we have on the x-axis the original sequence predicted minimum free energy, and on the y-axis is the improvement or change from the original MFE. Here, lower is better. And the blue dots are showing the predictions for our diffusion sampled sequences, and generally we see improved minimum free energy for these mRNAs. On the right, we have a very similar plot, but now for half-life, and we see a similar result where our diffusion sampled sequences are able to improve the predicted half-life. Further experiments have shown that we're not able to modulate the half-life with as much granularity as we are with minimum free energy. And if you're interested in this, definitely come check out our poster. And we're actively looking for interesting data sets that might help us with this task and collaborators.
Hi, everyone. My name is David Laub. I'm a PhD student at UC San Diego. I'm here to talk to you about my tool called GenVar Loader to apply sequence models to personalized genomes and tracks. For anyone who's not familiar with sequence models, they can, for example, be used to take DNA sequences input and then predict the read depth corresponding to RNA-seq or ATAC-seq experiments. Generally, they have only been trained on sequences from the reference genome, but recent work, many of which has even been talked about a few minutes ago, has shown that this approach really fails to capture the effects of genomic variants. And furthermore, that training on personalized genomes can really help. All right. So in order to do this, we'll need to change the reference genome and read depth to reflect those genomic variants. unfortunately, existing methods to do this generally, they were not thinking about training neural networks when they designed them. So for example, BCF tools requires writing individual genomes to disk. And if we scale this up to like thousands of individuals, we're looking at terabytes or petabytes of storage required just to generate this data. Instead, Genvar Loader is going to write those variants to a sparse memory mapped format and reconstruct those personalized genomes on the fly while also still including the indels. So as a result, Genvar Loader requires hundreds to thousands of times less storage than those FASTA files and enables training on thousands of individuals or more. So for example, the 1,000 Genomes project, there's 3,202 individuals in that. Writing all those FASTAs out would require over 6 terabytes of storage, whereas Genvar Loader requires just 3 gigabytes. And this is all while still maintaining a lot of performance. So Genvar Loader is over 1,000 times faster than reading the FASTA files, regardless of the size of the data set. In fact, it's so fast, it even exceeds the CPU to GPU transfer rate for state-of-the-art GPUs like NVIDIA A100s. And by also memory mapping track data, like from bigwigs, we get similar performance there as well. So what does this all mean is that during training, you can get really fast training speed-ups because you've eliminated all the data loading bottlenecks, right? So in this example, we're training a BPNet style architecture to predict personalized attack seek, and we're seeing a speed-up of about 3x. We've also put a lot of effort into making Genvar Loader as easy to use as it is performant. It only requires one new major step before you can get a PyTorch data loader ready to use. If you're interested in using personalized genomes or tracks in your research, come say hi at my poster. Thank you.
Recovering GRNs or gene directory networks using machine learning is like glorified curve fitting that doesn't actually fit. That was a joke to get your attention, but it does actually fit to some extent. I'm Zach. I'm working on perturb ODE and interpretable neural ODE for GRN discovery. It is well established through experiments in yeast and École that such regulatory systems are governed by nonlinear dynamical systems. In the simplest example, suppose gene X upregulates gene Y, then we can model the dynamics of gene Y as an ODE with a nonlinear activation of X and a linear decay of Y. By most other examples, the dynamics are much more complicated. That's why we're presenting our model, perturb ODE. Our objective is to recover GRN from interventional RNA-seq data and predict trajectories under unseen interventions. So in our data sets, we have hundreds of perturbations and a distribution corresponding to each intervention. Let's first look at our ODE formulation. So suppose we are given a gene expression vector. We first map it down linearly to a lower dimensional latent cofactor space with some additional shift and scaling. And then we apply a layer of nonlinear activation of a sigmoid function, and then we map it back up linearly using the B1 matrix. And our GRN is basically encoded by multiplying the B1 matrix and the B0 matrix with some additional scaling. And we also have a linear decay term. And we also have an intervention term that we set manually. If you look at the underlying parts of the ODE, you can basically think of it as a base vector field that is tunable like a neural network. And then we manually set the intervention so that this will lead to a different distribution of cells given different interventions. For training, for each intervention, we basically push forward some initial distributions to our prediction, and then we compare the Wasserstein-2 distance to our target, and then we backprop. Some additional comments: our model can manage both perfect and imperfect interventions, including both overexpression and knockout scenarios. And we also train with some diffused target densities to promote local stability by contraction mapping theorem. And we can handle dimensions as high as 1,000 with 32 gigabytes of memory. And this is our result. We outperforms DCDI, DCDFG, no tears LR in both real data and simulated data. And we are also capable of making accurate predictions for unseen interventions. So in conclusion, perturbable-D is a biologically realistic and scalable alternative to traditional SCMs. And if you're interested, please check out poster number 9. Thank you.